For the longest time, the way we fought cancer, well, it was like using a sledgehammer. But what if we could swap that out for something more like a laser-guided missile? That's exactly what we're talking about today. Let's dive into the revolutionary shift in cancer treatment known as molecular targeted therapy.
So why was this revolution so desperately needed? Well, just look at this number, 10 million. That's the estimated number of people who died from cancer in a single year, 2020. A number that staggering tells you one thing very clearly. We needed better, smarter weapons in this fight.
And this really gets right to the heart of it. On one side, you've got traditional chemotherapy, which just attacks all fast-growing cells, good and bad. On the other, you have targeted therapy, which is all about zeroing in on specific things that make cancer cells different. It's not just an upgrade, it's a completely different philosophy.
To really appreciate just how big a leap this was, we gotta take a quick look back at that older, much blunter way to fight.
Think about it like this. Chemo is a bulldozer. Its only job is to plow through and destroy any cell that divides quickly. The problem? It's not very smart. It can't tell the difference between a cancer cell and, say, a healthy cell in your hair follicles or the lining of your stomach. So it just bulldozes everything in its path, which is exactly why the side effects can be so widespread and so tough.
But the seed of a much, much smarter idea was actually planted over a century ago by a pretty visionary scientist.
This guy, Paul Ehrlich, way back in the 1890s, had this incredible concept he called a magic bullet. Imagine a drug that could hunt down a specific germ and kill only that germ without harming any of the healthy cells around it. Now, he was thinking about microbes, but that core idea, well, it became the guiding light for modern cancer therapy.
And this is where Ehrlich's dream starts to become a reality. For decades, it was just a cool idea. But then, things really started to happen. In 1998, we get a drug approved to target a protein called HER2 that drives some breast cancers. Then came the real game changer in 2001, imatinib. It turned a deadly form of leukemia into a manageable disease. That proved it could be done. The magic bullet was real. And from there, the floodgates just opened.
So how exactly do these modern-day magic bullets actually hit their mark with such precision?
The best way to picture it is a simple lock and key. Every cancer cell has specific molecules, let's call them locks, that it absolutely depends on to grow and survive. Targeted therapies are basically custom-designed keys that are built to fit only that specific lock. And when the key goes in and turns, it shuts the whole cancer operation down.
Here's what's happening inside that cancer cell. It's like a non-stop chain reaction, a line of dominoes falling one after the other. And every domino that falls sends the exact same message down the line. Grow, divide, survive, and do it all again, completely out of control. And here's the solution. The drug is like reaching in and pulling one crucial domino right out of that line. It blocks the key signal. The chain reaction is broken, the messages stop getting through, and the cancer cell's growth engine just grinds to a halt. Now, these keys come in two main styles. You have what are called small molecule inhibitors. These guys are tiny, small enough to actually sneak inside the cancer cell and jam the lock from within. Then you have monoclonal antibodies. These are the big guys. They're too large to get in, so they work on the outside, clamping onto the lock so the signal can't get in the first place.
But of course, the story doesn't just end there with a miracle cure. You see, cancer is a formidable enemy. It's smart, it's adaptable, and it learned how to fight back. And this fight is all about something called drug resistance. It comes in two main flavors. First, there's primary resistance. That's when the drug just doesn't work from day one, for some reason. But what's more common and maybe more frustrating is acquired resistance. That's when the drug works great for a while, but then the cancer evolves. It figures out a way around the drug, and it just stops working.
So how in the world does it do that? Well, it has a few really clever tricks. Sometimes it just mutates its target. It literally changes the lock so our custom-made key doesn't fit anymore. Other times, it finds a detour. It just activates a totally different set of signals to keep itself growing, bypassing the roadblock we put up. Or sometimes a few rare cells that were already resistant to the drug survive the initial treatment, and with all their competition gone, they're free to multiply and take over. Okay, so when you put it all together, the incredible promise, but also these huge challenges, where does that leave us today? Well, this is the reality of what we call precision medicine. So here's the deal, the pros and cons. The pros are just revolutionary. These therapies are often way more effective with far fewer of the brutal side effects of chemo. This is personalized medicine. But the cons, they're very real.
Drug resistance is a constant battle. This means for most people, these drugs aren't a cure. And the biggest catch of all, they only work if your particular cancer has the specific molecular lock that we've managed to build a key for.
There is no doubt that targeted therapy completely changed the game. For so many people, it transformed cancer from an almost certain death sentence into a manageable, chronic disease. It proved that Paul Ehlers' magic bullet was actually possible. The question now, as we're in this constant cat-and-mouse game with drug resistance, is what will that next great leap forward look like?